So let's start this session with David Johnson. Please go ahead.
Oh, okay. That was quick. Okay. So yeah, first I just want to thank the organizers for giving me this opportunity to come back here. I was indeed here 8 years ago. I'm sure my hair is exactly the same color, but it was super enjoyable last time, and it's definitely good to be back. So yeah, what I'm going to talk about today is basically what I'm going to talk about today is something that has never been publicly vetted. It's all in preparation. This is some ongoing work. That's one of the chapters of Yin Ma's dissertation, who's a PhD student with me that will be graduating quite soon. So really, Yin deserves all the credit for everything I present today. It's been fantastic working with her, and it's going to be sad to see her leave pretty soon. And then she received help from Joseph Ramoneda, who was a postdoc in my lab for a while and now at the University of Colorado. He did a lot of the statistical analyses that I'll show you today, and Anton Kahn at ETH, who assisted us with some of the individual-based modeling that I'll show you today. So we think plasmids are pretty cool. I don't think we really need to introduce what plasmids are. You guys probably already know. I just got to press this button and see if, yes, it does. But they're pretty cool in that they encode machinery that helps with both their vertical gene transfer and their horizontal gene transfer. So of course, they're little circular pieces of DNA that are extra chromosomal. They have their own origin and replication. They contain machinery that enables their transfer to other species. And what is quite interesting is they also carry cargo genes that provide context-dependent benefits to the host. Of course, antimicrobial resistance and xenobiotic degradation are well known, so that it can also ensure or improve their vertical transmission. So I think it's extremely elegant that you have such a small system that enables both VGT and HGT in an amazingly effective manner. Our main interest in this project was to try and understand the fate of these plasmids in biofilms. So we have a biofilm community with a number of different species or genotypes or whatever you want to call them, where some of them contain the plasmid. And as you guys know, this is an incredibly dynamic process. So several processes can be going on at the same time in these biofilms. For instance, you can have plasmid transfer between carriers to new species that did not previously contain the plasmid. You can also have plasmid curing and loss events due to errors in the segregation control system in these plasmids. So when they divide, some of the daughter cells can emerge that are now plasmid free. And then, of course, in the absence of selection for the plasmids often carry costs, which can lead to competition between plasmid carriers and plasmid free, where the plasmid free often have a growth benefit compared to their carriers, leading to the proliferation of plasmid-free versions in the absence of selection. So what I want to talk about, though, is that we have this spatial arrangement or spatial self-organization within a biofilm, and how does that direct the fate of that plasmid if we then impose positive selection for the plasmid? So for example, if I go in and add antibiotics, for instance, to say this is antibiotic resistance encoding plasmid, I apply antibiotics, I will destroy or remove a lot of the sensitive cells, leaving little patches of those that carry resistance, or perhaps are located and physically protected from the antibiotic for one reason or another, whether that's degradation of the antibiotic or just being in a state that is dormant and insensitive to the antibiotic, whatever. And then after you then remove the antibiotic, then the community proliferates, and because the composition has changed, you reach a new pattern of spatial self-organization. And so we kind of want to try to understand these dynamics of how spatial self-organization, distribution of the plasmid in space and between species, then influences the effects of the community upon positive selection. This then changes the spatial organization again, which then leads to regrowth. So it's a highly dynamic process. And then, perhaps obvious, but this initial state of spatial self-organization is obviously going to have large effects on these dynamics coming down here. So if I start with a totally different spatial self-organization, so community composition in these two cases is identical. It is a little bit embarrassing that I actually counted out the dots to make sure they are identical. The plasmid dosage is identical, but just the spatial arrangement differs. And then, of course, if you apply the antibiotics to this, you can have a very different outcome after application of the antibiotics, and then a very different community upon regrowth. And so this is kind of what we're interested in, is how do these initial states of spatial self-organization dictate the effects of antibiotics and regrowth after removing the antibiotics. So I don't want to put up slides about the gut microbiome, but for example, if I were to have a healthy gut microbiome and I take antibiotics, I'm going to change my community, and then after stopping treatment, I will get regrowth. And so these are kind of the ideas that we're thinking about. Okay. Now the challenge here, though, is that spatial self-organization itself in these states is a very dynamic process in many cases also. I guess not in these anaerobic methane oxidizing, then you have a pretty stable spatial self-organization, but I think in many systems, it's actually an incredibly dynamic process. And I think the easiest way to show this is just with experiments. So this is an experiment we did. We basically have two types, a donor and recipient. They are genetically and phenotypically identical, except they express different fluorescent proteins, and the donor contains a plasmid. And this plasmid that can transfer to the recipient and become a transconjugate, which has a different color. And I just want to show you guys this process of spatial self-organization during growth. What Yin did here is she just mixed the donor with the recipient. She grew them separately in test tubes, mixed them 50-50, and put a drop on an agar plate. And so these are the initial conditions. And then what I'm going to show you next, it's not a time lapse. I should be careful. All I'm going to do is expand you the view so you can see how this colony develops. Now, the reason this works is because resources are emerging from the outside, coming in, and because resources from the outside are coming in from the periphery, only a thin layer of cells at the expansion frontier is really contributing to further growth. And so it's imperfect, but this basically provides a historical record of what happened as this colony grew. Okay? But what I want to show you here, just to emphasize the complexity of these types of dynamics, in this case, what we did is we allowed growth to start, the community expands outwards, and then we hit it with antibiotics, and you can see the patterns totally change. I can zoom in here and show you. So what we do is this is that initial drop where the magenta and green are well mixed together. And what we see is as they expand outwards, this magenta actually loses the plasmid, reverts back to red. But then when we hit it with antibiotics, we now recover some of those magenta that take off, and these cyans start to proliferate. And so based on this image, the question I want to pose is maybe a bit practical, but is there an optimal time to apply antibiotics during this range expansion process to minimize the proliferation of these transconjugates? Can I make basic predictions about when it's best to apply antibiotics and hit the system to prevent these guys from proliferating. So for instance, you can imagine this recipient as a potential pathogen, and we really want to minimize this transfer over here and proliferation of these guys. So this is kind of the question we were hoping to address is can we get at this? So, you know, my initial intuitions are almost always wrong. But I initially thought that we should observe a negative relationship. So basically, the amount of transconjugants at the frontier of this expanding colony after applying antibiotics, we thought it should be negative related to the time that we apply antibiotics. So if you wait as long as possible before applying antibiotics, then the transconjugants at the frontier should decrease. And we thought this for two reasons. First, we know that drift at the expansion frontier, these expanding colonies, reduces intermixing of the pattern formations that we see. And I'll just show you this real quickly. Here we have different colonies that these are just different experiments. We have two competing types with the plasmid, then we do plasmid swaps. Here they're metabolically interacting through cross-feeding. It doesn't really matter, but what we're doing is we're quantifying the amount of intermixing starting at the frontier, the edge of that initial drop, outwards as it grows. And what we see is the amount of intermixing, that's intermixing between the two types, it rapidly decreases as you move outwards from the founding zone to the edge. And this occurs regardless of what type of metabolic interactions you impose. It doesn't really matter, but we generally always see a decrease in intermixing as a colony gets bigger. And this is well known by work from Oskar Holacek and David Nelson, why this is. So as a colony expands outwards, this is some experimental data from our own group, that these boundaries between two competing types, they have wobbles to them. So in this case, you know, the competing types is green and blue. They should have identical growth rates, so they expand outwards in the same velocity, and then these boundaries emerge that are parallel or perpendicular to the axis of expansion because both types have exactly the same velocity. But what happens though is that these boundaries have wobbles and wiggles on them just due to stochastic processes, and that can cause boundary coalescence events, such as right here in the blue. So two green, their boundaries just wobble together and collide. Here they collide over here. And here there's lots and lots of collisions going on right at the beginning. But what this does is it gradually deteriorates intermixing as you expand outwards. So the idea is that this is just a stochastic process. And the consequence is you have reduced intermixing. Now, of course, reduced intermixing means you have less cell-cell contacts between two different types, right? So if you're reducing intermixing here, obviously the number of cell-cell contacts right at this edge between blue and green is really, really high. But as intermixing decays, over here, the number of cell-cell contacts between blue and green decreases. And for plasmid conjugation, which is, of course, a cell contact dependent process, it requires physical proximity or even contact between two different cell types. And so, yeah, one would imagine that the amount of plasmid transfer between, say, blue and green would be much, much lower in a system like this, where you have very little intermixing, versus a system like this, where they are highly intermixed. It's basically just saying that plasmid transfer should be proportional to the number of cell-cell contacts. And the number of cell-cell contacts decreases over time due to drift at the expansion frontier in these expanding colonies. And then the other reason we expect a negative relationship is simply because plasmids are costly in the absence of selection. So if I take two types, plasmid-free and a plasmid carrier, then we would expect the growth rate of the plasmid-free to be higher than the carrier. So this is just experimental data from our group using another method by Oscar Holacek and David Nelson, where you can quantify the relative growth rates of a plasmid-free and plasmid carrier simply by colliding them on space and taking into account the geometry of the collision boundary. Then one can estimate the cost and growth rate of carriage of this plasmid. And in our case, we see about a 17% reduction in growth rate when a plasmid-free cell carries the plasmid. And of course, if a plasmid-free cell is growing faster than a plasmid carrier, then during the expansion process, they will start to displace the slower growing organism. And so you can see that here, if green's growing faster than red, and that means that the boundaries between green and red are no longer straight. Straight boundaries would give an indication of equal growth rates, but they start to curve outwards. And this can mechanically like pinch off a red, which then essentially drives it to extinction because growth is just confined to the thin layer at the front. So if you sum these together, during colony expansion, we have a decay in intermixing, so we have reduced opportunities for plasmid transfer between a recipient and a donor. And we also have a reduced plasmid load. So the plasmid load's going to decay also purely because plasmid-free individuals grow faster than plasmid carriage. So if you sum these together, that's basically why we came up with a negative relationship. So what that means is that if you apply antibiotics at early times, we expect that to maximize the number of transconjugates because you still have a lot of intermixing and you have a high plasmid load. But as you wait longer and longer, your intermixing deteriorates, your plasmid load decreases, and then if you apply antibiotics at later times, then you get fewer transconjugates at the frontier. So this was our intuition. So as a first step, though, we just wanted to verify that intermixing and plasmid load do indeed decline during the range expansion process. So this is just doing expansion of a plasmid-free and a plasmid carrier in the absence of selection. And so to do this, our experimental approach, I've already kind of introduced it. We have a donor cell type and a potential recipient that are otherwise phenotypically and genetically identical, except they express different fluorescent proteins, so they are peer competitors. Into the donor, we put in a plasmid that confers chloramphenicol resistance and encodes a blue fluorescent protein. So it appears, it looks magenta on my computer, but I guess purple. If the plasmid transfers to the potential recipient, the transconjugate then becomes the composite color green and blue, so it appears cyan. And then the third thing that can happen is as the donor divides, it can be cured of the plasmid and return back to its red state. So that's our simple experimental system. The next slides are more philosophical. Oh wait, sorry, not yet. So all we do is we put a 1-to-1 ratio on a plate. All we do is we simply wait 7 days and then we image them under microscopy. So the next is a bit philosophical. I used to call myself a bottom-up experimentalist, and I would say that this is synthetic ecology. After talking to some people here over the past few days and reflecting a bit, I started working with a theoretician, Stefan Spatial from the University of Bern, who's a population geneticist, and he's interested in our range expansions, applying population genetics models to them. But anyway, we would basically do experiments, and I'd send him an email, and I would send him the email, be like, here's some new experimental data for you. And he'd be like, oh, great, that's fantastic. And after some time, he didn't mean this in a derogatory sense in any way, But I discovered that when I would send him experimental data, he did not call it experimental. He said that what we were sending him were wet simulations. And at first, as an experimentalist, I was a little bit offended. But I see the point, and I think it's just something that in general in the field of synthetic ecology we need to be careful about. I think synthetic ecology is incredibly powerful for identifying principles of how systems work, but and it's good for validating models, but I think we should be very careful about how much they actually reflect or how much we can learn about real community functioning. But to me, that's the grand challenge is, what principles that we identify with synthetic ecology continue to hold in more complex systems. And that's why I no longer view it as negative. I've fully adopted it, and I'm fine with it. And that's the position I'm at. Anyway. So anyway, our wet simulations. So as I mentioned, we just mix these two together. We put them on a plate. We let them expand. And this is kind of what we see. So this is that initial colony. In the absence of selection, it grows and expands. And what you'll see here is right at the very beginning, we still have some transconjugates, and we have plasmid carriers, and they're very rapidly displaced by the cured donor and the potential recipient that never received the plasmid. The main point here, though, is that, yes, indeed, we get a very sharp decline in the plasmid load over time. And so based on that, back to our initial expectation, that was our initial thought, is that when plasmid load is high here, if you hit with antibiotics at this point, when the plasmid load is high, you'll have a lot of transconjugates at the frontier. But if you wait long enough and get these purged out just through natural selection, then you should have less transconjugates at the frontier. So then we started applying antibiotics at different points in time. So we applied antibiotics at these different times. Total of 8 different times. So just so you can see where it would lie on this plasmid load curve, we clustered quite a bit of points right at the early phase and then spread them out. And then the experiments we ran are pretty simple. So in one case, we have replicate plates where we apply antibiotics basically right at the beginning, and then we wait 7 days in image. And then at the other extreme, we wait 108 hours, and then we apply antibiotics and image, and then we have a number of data points in between with various amounts of time that we wait. And this is what we observe experimentally. So what you can see is if we add antibiotics immediately, we are dominated by the donor. And as you wait longer and longer, you get an increase in the number of transconjugates coming up. But then if you wait a really long time, it collapses back down where the number of transconjugates reduces. So we don't get a monotonic decreasing relationship. We get a unimodal type behavior where the amount of transconjugates at the frontier peaks at some intermediate treatment time. So then the question is why? What could give rise to such a unimodal relationship? And so to do that, we switched over to, I guess, dry simulations. We're using an individual-based model. The model we use is Cell Modeler, which is an individual-based framework. It really works really good for us because we can model tens to thousands of cells, and it really takes into account the biophysical processes acting on cells. For example, it considers cell morphology, so it takes into account the rod-shaped nature of some cells, and really nicely, it can account for contact-dependent processes, so processes that occur when two cells come into physical contact with each other. This came out of Jim Hasselhoff's lab here at Cambridge, and it's been very effective for us. And so just to go back, I'll show you the experimental data again, and here's our simulations. So again, what we did is we applied this experimentally measured plasmid costs, and then we knew in our system that the plasmid loss probability is much higher than the plasmid transfer probability. So we toyed around with this to basically eyeball match what we see experimentally. And you can see that doing this, we can get simulations that match our experiments quite well. So at time equals 0, our simulations do indeed show that it's dominated by the donor, and that at very late times, then you only get small patches of the donor persisting. But the cool thing here, though, is that as we saw experimentally, with our dry simulations, we also get this monotonic behavior in the amount of the transconjugate, where it seems to peak at intermediate to antibiotic administration times. So that's from our simulations. We get this nice unimodal relationship similar to our experiments. So again, though, why is this? I'll just switch through this. So we do lineage tracking too, but I'm going to run out of time. So to do this, Yinyan's hypothesis is that it's really an interplay between these two plasmid loss and plasmid transfer rates that are giving rise to this unimodal relationship. So when she had set the plasmid loss and plasmid transfer rates to 0.005 and 2, she gets that nice unimodal relationship. When she then turns the plasmid loss rate down, she can actually generate scenarios where we get the exact opposite of our initial expectation, where we actually get an increasing relationship. And the idea here is that the plasmid transfer rate is sufficiently higher than the plasmid loss rate and the consequences of the cost that the plasmid can still continue to spread. And then we continue to get transconjugates that increase monotonically positive the longer and longer we wait to apply antibiotics. And then, of course, you can switch this, and we can set the plasmid loss rate to be much, much higher than the plasmid transfer rate. And this looks flat. It's actually statistically monotonically decreasing like we initially expected. And here it makes sense because in this case, the plasmid is lost so rapidly that it can't keep up with the transfer, and it will decline and disappear from the system. So we can go through this more systematically and examine the entire space of probability of plasmid loss and plasmid transfer. And indeed, you can get any possible outcome depending on these two parameters. So when plasmid transfer rates are high relative to plasmid loss, you're dominated by these monotonically increasing relationships. Down here, when plasmid loss rate is high, relative to plasmid transfer, then you can get monotonically decreasing. Here they're flat because you basically just kill everyone right away and it doesn't really matter. And then that sweet spot in between, this transition point where we get these unimodal relationships. And I'm supposed to be done. I am done. I'll just quickly say what we're doing now. So we're now interested in trying to extend this into patchy biofilms. So the idea is that, very often you have patchy biofilms with small discrete areas that over time will grow and they will coalesce into a larger biofilm. And of course, the time for this patch to collide into this patch, it's also a timing thing, right? So this is a long period of time before they collide. This is a short period of time before they collide. And we're seeing this as an interesting opportunity because we view these collision boundaries between, say this is a plasmid carrier and a plasmid free, but those collision boundaries will then be hotspots for plasmid transfer. And so you can see that over here. This is a potential recipient that doesn't have the plasmid. This is two plasmid carriers. And when the colonies collide, they have this contact line where you see plasmid transfer going into the recipient. But of course, plasmid loss is also occurring here, and then those areas become dark. And what that means is that the timing between collisions of discrete biofilm units will have major impacts on the spread of resistance. So here you have early collisions and you get a lot of transfer, but you have slightly later collisions and it rapidly disappears. with that, since I'm leaving very little time for questions, I just want to acknowledge again, Yin deserves all the credit for this. It was really her brainchild, and she did all the experiments, and also to Josip and Anton. And then I also just want to thank my group. I feel incredibly blessed to be working with such a fantastic group, and thank you all for your time.
Thank you for this nice talk. Since we're a little late, we still have time for a few questions. There's one there, Orgon.
Thanks for your nice talk. Can you comment on the effect of motility or swarming?
No, I can't, but we have been starting these experiments. We do have preliminary data where we're varying it under agar concentrations. In these experiments here, there's no active motility. They're just pushing and shoving each other around. But if we lower the agar concentration, then we can add motility. And of course, this has big effects, for instance, on the effects of drift at the expansion frontiers. It will have very large effects. And whether we would get qualitatively different outcomes, I don't know. But it is something that we're starting.
So is Mike in the room? If she is, then start setting up while we take a question more.
I would want to have so if you're in this intermediate in the sweet spot regime, what could we translate it to hypothesized state of gut? You think that it would be ideal to run the system into a strong dysbiosis and then hit? Then you have the abundance without the plasmid and then hit?
I mean, based on the unimodal, you would have two choices, right? You hit it as quickly as possible, you wait as long as possible. But that window where it's increasing during the unimodal, I don't know how short that is. I mean, we have an interesting plasmid that has a very large plasmid loss rate. It's probably, this plasmid is not stable in our organism. And so I think it really depends on that plasmid loss rate, but it's possible that window is so short that trying to catch it is virtually impossible. Yeah.
There's one last question now.
A practical question. How do you make sure that when you deliver the antibiotic on the plates at different times, the antibiotic is all well spread uniformly on the whole plate?
My hearing really sucks too.
So, when you deliver the antibiotic to the plates,
Yeah.
how do you do that, ensuring that it's all well spread uniformly around the plate?
It's an approximation, so we just put 4 drops spaced in quadrants around it. So there is going to be some diffusion limitation. So you will have, initially it will not be truly uniform, but I think you obtain something close to uniform quite quickly.
Okay, let's thank the speaker again. Our next speaker is Mike Sangster from Grenoble.
Oh, thank you.